Okay, thank you for the introduction. My name is Wei Zhang. I'm a senior scientist at Goldfinch Bio Company, and I was a postdoc in Dr. Triadker's lab in UC San Diego. So today I'll talk about this project which I conducted in Dr. Adker's lab. So cancer is a heterogeneous disease, and to understand the heterogeneity, it is a very popular approach to aggregate the information of tumor mutations in the context of molecular networks. Many algorithms were developed on this basis, and the heart of those algorithms is network propagation. And here is how it works. Suppose we are given a molecular network where each node is a gene and each edge is a gene-to-gene interaction, and we are projecting two patients' mutation profiles onto this map. So patient 1 has the mutations on the blue genes and patient 2 has the mutations on the yellow genes. At the first glance, these two patients are very different in terms of their mutational profile, right? So network propagation works like a heat diffusion. So the concept is to propagate or diffuse the information of the tumor mutations across the network edges, such that at convergence, the score on each node represents the network distance to the original mutation. So we can see from this figure that after network propagation, it becomes apparent that there is a network region which is repeatedly targeted by the mutations in the two patients, and such information can be very useful in grouping the two patients into the same subtype. So although this type of algorithm is promising, it has some open challenges, such as the choice of networks, the choice of interaction types, and specifically for cancer study, the reference molecular network are neither tissue-specific nor cancer-specific. So this may lead to the erroneous aggregation of passenger mutations, as well as the contamination of characteristic pathway signals. So it is very important to distinguish between the interactions that are functional in tumor cells and those that are not. To address those questions, we developed an algorithm called network-based supervised stratification, which is on my GitHub. So this method extends the existing supervised random walk algorithm with a novel loss function designed specifically for cancer subtype classification. So like many of the previous similar methods, this algorithm also uses a network propagation, which is based on a random walk with restart model. So this model takes into consideration two pieces of input informations. The first information is a tumor by gene binary matrix, which represents the mutation profiles for each tumor. And the second piece of information is a gene by gene degree normalized adjacency matrix, which represents the molecular network. So random walk works like iteratively multiplying this P matrix against the Q matrix. And with a small probability of restart, the random walk will restart at the initial state P0, which is the binary matrix. So this equation was applied iteratively until it converges at a state where PT plus 1 equals to PT, representing the network propagated mutation profile for each tumor. Based on this model, we added an additional goal to learn a weighted transition matrix such that at convergence, the network propagated mutation profiles for each tumor better represent some predefined tumor subtypes. And below, I will introduce the workflow of our algorithm. So one of the key differences between the supervised version of random walk and the unsupervised version of random walk is that here, each edge is annotated by a set of edge features, and those features can include the types of interactions, the source databases, or the mutual exclusivity of the two genes connected by the edge, etc., etc. So these edge features, parameterized by a vector of feature weights W, were then summarized using a sigmoid function to compute a single activation score for each edge. The result is an activation score between 0 and 1. So 0 means that the edge is practically removed from the network propagation. And 1 means that the random walk is very likely to traverse through this edge. Having computed an activation score for each edge, we then plugged in those edge features to compute a weighted transition matrix. And based on this weighted matrix, we propagate the mutation profiles until convergence to calculate this propagated mutation profile. And since our algorithm is supervised learning, here we are providing another piece of input information, which is the subtype labels for each tumor, such that for each tumor U, we can calculate the distance we can calculate the true subtype center CA and the nearest neighboring subtype center of tumor U, CB. So to formulate a cost function, we introduce this term du, which basically captures the distance of tumor U to the true subtype center and the distance of tumor U to the nearest neighboring subtype center. The intuition is that we want the first term of du to be small and the second term of du to be large. In that case, we know that the tumor U is close to its true subtype center and far away from the neighboring subtype centers, which basically means that the classification is correct. So we finalize our optimization problem like follows. So the objective function have 2 terms. The first term is an L1 norm, which basically controls the complexity of the model. And the second term is a logistic transformation of this term du, which gives a curve like that. So the intuition is that if the first term of du is small and the second term of du is large, which, as I said, represents tumor U is correctly classified, then the cost function with respect to tumor U will be close to 0. In the contrast, if the first term of du is large and the second term of du is small, then it means the classification of tumor U is terribly wrong, which gives a cost function with respect to tumor U close to 1. So that you can see that here we are basically translating the classification error into a cost function. And here, this cost function is differentiable so that we can use gradient descent to minimize the cost function. So to summarize, here is the entire workflow. It starts with calculating a single activation score for each edge, and then calculate a weighted transition matrix, propagate the mutation profiles, and calculate a cost function. And if it is not converged, we calculate the gradient descent using the chain rule to update the edge feature weight W. So this algorithm was applied iteratively until the cost function stops to improve. To better demonstrate our algorithm, we first performed a simulation. So the simulated data contains 100 tumors and 1,000 genes. The tumors were divided into 2 equal-sized subtypes. So our goal was to recover these 2 subtypes, and as well as to review the underlying subnetworks of these 2 subtypes. So each of the 2 subtypes was assigned with 50 characteristic genes, and for each tumor, a unique characteristic gene was mutated. And we are also randomizing some random mutations as well as a high-frequency mutation, such high-frequency mutated gene, such as gene number 16 on this map. So the edges were randomly generated with a higher probability among all the characteristic genes and lower probability among the other genes. We first evaluated the performance of the unsupervised version of random walk. In this case, all the edges are equally activated, so the random walk have equal probability to traverse through all the edges, given it's on a specific node. Since all the characteristic genes were highly interconnected, both within the same subtype and between the 2 different subtypes, the final propagated mutation profile, the final propagated mutation scores were evenly distributed among all these genes, and as a result, the propagated mutation profiles failed to recognize the 2 predefined subtypes. Next, in order to perform the supervised version of random walk, here we are providing some subtype characteristic edge features, which were set to 1 if they are connecting 2 characteristic genes of the same subtype and 0 otherwise. We are also providing some random edge features for the training. So when plugged in those edge features and perform the supervised version of network propagation, our algorithm soon realized the underlying fact that these subtype characteristic edge features are very important. Therefore, it assigned very high positive weights to those features. So consequently, two subnetworks emerge with the boosted edge activation scores underlying. So these 2 subtypes are characterizing the 2 predefined subtypes. And as a result, the propagated mutation profiles better delineate the 2 predefined subtypes. And overall, our supervised network propagation achieved nearly perfect classification of the 2 predefined subtypes, whereas the unsupervised version of network propagation failed to delineate the 2 predefined subtypes. Finally, we applied our algorithm on a real data set, which the task is to classify the breast cancer tumors. So basically, we used 863 tumors from TCGA, which were then divided into a training set and a validation set. And these tumors were predefined as 4 subtypes: basal, HER2, luminal A, and luminal B, based on gene expression and clinical data. So in this task, we use 76 edge features, and we are using a pathway commons network as a reference network. We first evaluated the performance of the training dataset. So this is a learning curve where x-axis represents the number of iterations, and y-axis represents either the value of the cost function or the accuracy. So we noticed that there is a strong anti-correlation between the cost and the accuracy, which is a desired feature and basically confirms that our cost function translates well to the goal of the accurate classification. And the training accuracy starts with 55% at iteration 0, and it increased to a plateau at about 70% at around iteration 200. Then we evaluate the performance of the validation dataset. We can see the validation accuracy also increased from 53% to something between 66 to 68%. And this performance is much better than the baseline performance achieved by using either the mutation profiles without random walk or using the unsupervised version of random walk. So we also visualized the performance of the classification using this confusion matrix. And in this table, each row is the predefined subtype, and each column is our predicted subtype. We can see that in 3 out of the 4 subtypes, the performance is pretty high. The 3 subtypes are basal, HER2, and luminal A, whereas in luminal B, the performance is a bit lower, which is understandable because it is known that luminal B is the most heterogeneous in terms of its mutational profiles. At convergence, the weighted network was of particular interest as it reveals the underlying subnetworks of the breast cancer subtypes. For instance, there is a 10-gene subnetwork underlying the luminal A subtype centered on PIK3CA, CDH1, cyclin D1, and NOTCH1. These 10 genes were highly interconnected, indicating they are functionally linked in luminal A breast cancers. And the second subnetwork of interest, characterizing both basal and HER2 subtype, is centered on P53, EGFR, NOT4, and MET. Interestingly, our algorithm also learned the fact that many interactions should be removed from the network propagation to avoid the contamination of the subtype-specific mutation signatures, such as this edge between P53 and PIK3CA. That is because the algorithm learned the fact from the training data sets that the mutations on PIK3CA is enriched in basal and HER2 subtype, whereas it is depleted in luminal A subtype. In the contrast, the mutations on PIK3CA is enriched in luminal A subtype. So if the network propagation was allowed through this edge, then these subtype-specific mutation signatures will be contaminated with each other. To summarize, here we developed an algorithm which iteratively performs the weighted network propagation, the supervised tumor classification, and updates the edge activation scores accordingly. So the supervised learning is used to adjust the weight of each molecular interaction, guiding the direction of network propagation to maximize the agreement of tumors of the same subtype. I want to emphasize the cartoon that I showed early on in this talk. So the random walk is like a person on this very complex roadmap, right? So the person kind of like rely on the information on the map or the information on the signpost to determine which way to go. But if the information was so overwhelming and it is sometimes irrelevant to his task, then the person will get lost, right? So consider that if the person is not holding a map, but instead a GPS device, then the algorithm will summarize all the information on the map, and based on the destination, it will calculate the best route to take. That is basically analogous to our algorithm. So for this, I would like to thank my PI, Trey Edker, who conceived this study and is very supportive to this project and to many my other projects that I conducted in his lab. And I want to introduce his keynote talk at 5 p.m. also in this room. And I would like to thank my collaborator Jianzhu Ma, who is an expert in mathematics and machine learning, and he helped me a lot in developing this algorithm. So thank you all for your attention.
Okay, we have time for a couple questions.
Yeah, nice work. I wonder in your performance, and you show that it's not really 100%. We know that nothing is 100%, but given the example you shown that there are really pathological classification, luminal A is tend to be ER positive and B is also ER positive, and then HER2 is ER negative and HER2. Have you actually looked at the signatures and see whether you could actually take advantage of those biological knowledge or the lack of biological knowledge was the reason that the performance was not really closer to 90 or 100%?
Oh, okay, thank you for your question. So yeah, the straight answer is no, we didn't take into consideration of those clinical data to in training the model. So yeah, I guess by including those in the model, it may increase the performance. But here I want to emphasize that the goal of this work is to bridge the link between the tumor mutations and the clinical subtypes predefined by either the clinical data or the gene expression. So we want to find the mutational causes of those subtypes. So if that makes sense.
Great, thank you very much.
Okay, thank you.
Okay, our next talk is by Shuza Li on multi-omics, multi-scale networks of human immune responses to vaccination and infection. And he is from Emory University.